Okay, Unit B. Here we go. Unit B, enzymes. We're going to be talking about enzymes. So what are enzymes? Remember in the last unit we talked about proteins and proteins made of amino acids. Enzymes are proteins. They're functional proteins. That's what they are. They're a sequence of amino acids that has tertiary or quaternary structure. So we got a lot of hydrogen bonds, among other things, happening to make complex 3D shapes. And what enzymes do is enzymes carry out chemical reactions. They catalyze chemical reactions. They're a catalyst to chemical reactions in your body, metabolic reactions in your body. What does that mean, catalyze, as a catalyst? What is a catalyst? A catalyst is something that increases the likelihood or aids the likelihood of a chemical reaction occurring. Now, in our bodies, basically what enzymes essentially, they do a lot of things, but essentially what they're doing is usually they are breaking bonds or combining bonds together or putting things together. Breaking things or putting them together. When you break things apart, that's called catabolism. Cat, because cats scratch stuff into little pieces, so that's catabolism. And when you put things together, that's called annabolism. Okay? Catabolic, anabolic reactions. Catabolic, breaking things down. Anabolic, putting things together. That's what enzymes do. So enzymes are building, so when you grow, that's enzymes building stuff up in your body. When you digest food, you break it down for energy, that's enzymes breaking it down. Everything that's happening in your body is being mediated and carried out, every reaction is being an enzyme is a catalyst for that. So what does it mean to be a catalyst? So it means that you help a reaction occur and you don't get used up in that reaction. You're not a participant in that reaction in terms that you are not contributing, you're not a product. So in an enzyme, in an enzyme in a reaction where an enzyme is a catalyst, you will have a substrate that will then be converted into a product or products. Or you could have substrates that are converted into a product, right? So you could have substrates that are converted, a bond is made into a product, or a substrate converted into products. Okay, so let's talk about enzymes. So what enzymes do in our bodies, what enzymes do, or how they act as catalysts, is there are these three-dimensional molecular structures. And what enzymes have, the most important part of an enzyme, so you imagine you've got that quaternary or tertiary structure of a protein. So you've got some complex shape. Imagine my hands are this complex shape. And the enzymes will have an active site, an area, a region on the enzyme where the R groups, remember the R groups, the amino acid R groups, are coming together and their chemical properties are going to influence a chemical reaction, are going to do something. The other thing to note is this shape is very important in an enzyme because that shape, those R groups, the shape of those R groups, the shape of that enzyme is specific to a substrate. So an enzyme acts on a specific substrate. They don't just grab anything, they have a specific substrate. So for example, I have an enzyme here and it's a specific shape that the substrate is this marker. So in your body, that could be an enzyme, red substrate is a sugar, it's glucose. Okay? And this substrate, so when a substrate and an enzyme, a substrate enters they're mowing the lawn out there, great. When a substrate enters the active site, that's called an enzyme substrate complex. Enzymes are usually named for their substrate, not for their product, for their substrate. So, and then you just add ACE, an ending of ACE. So this would be, what is this? This is highlighter. So this would be highlighter ACE, highlight ACE, highlight ACE, highlighter ACE, something like that. Not all the time. It's typically, but there are some enzymes that are named, the names don't make any sense. The example, what is the example that we use? Maltose, so an enzyme that works on maltose that breaks down the disaccharide maltose. Remember disaccharide from Unit A? That breaks down the disaccharide into two monosaccharides of glucose and fructose is called maltase. Okay? So, how does an enzyme work? What happens here? So let's say we have a substrate. It is dihighlighter, okay? Two highlighters together. This makes a it's not a disaccharide, but a dihighlighteride. And the enzyme, we're gonna say my hand is the enzyme. And so it's got an active site that is specific to this highlighter. Now, how enzymes work, the big advantage of enzymes is they lower the activation energy of a reaction. They lower the activation energy. What that means is chemical reactions need energy put into them often to get them going, to start the reaction. So if you're joining things together, they need to have enough energy to smash into each other. That's why oftentimes you need temperature for chemical reactions to happen, because what temperature does is the molecules start moving faster. Temperature is just molecules moving faster, banging into each other. And so imagine these were two molecules in a solution, and we're heating it up, and now they're banging together, and eventually, by chance, they would bang together and form a new substrate. That's what chemical reactions do, and a bond would form here, an ionic bond or a covalent bond. For metabolic pathways, for what we're talking about, there's going to be covalent bonds. Okay, it's often when we're joining things together, we're making covalent bonds. We need enzymes to make covalent bonds. So for biological systems, this is going to be covalent bonds. So this can happen spontaneously if there's enough energy. It's really hot, and these things are banging together with enough force that it makes this dual highlighter. What an enzyme does is it lowers the activation energy. So it doesn't have to be so hot in here. These things are just floating around, and they don't, when it's hot, they're just randomly slamming into each other. But what an enzyme can do is it comes along, the active site, which is specific to the substrate. So this hand is specific to, so this is one molecule. You gotta imagine this is one molecule, and so they're floating around. I wish I could make them float around. Here, I can. Here, so let's say they're just floating around, and the enzyme is floating around, and so this one grabs this one grabs this, and by grabbing them, what happens is the enzyme puts them together and lets it go. That's an enzyme reaction. That was an anabolic reaction, because we took two smaller things and we put them to put bigger things. Now notice, they didn't have to be moving as fast. The enzyme orients them physically correctly, so they merge together correctly. And the activation energy is a lot lower. So this is beneficial to organic systems because we don't have to be on fire for chemical reactions to happen, because essentially if you go to a chemistry lab, everything is always heated on a hot plate and on fire for chemical reactions to happen, or next to being on fire. Enzymes make it, because the activation energy is so much lower, we're not on fire. So now we can go the other way. Oftentimes these reactions are reversible, but especially these anabolic, catabolic. So then the enzyme can grab the active site, the substrate, this marker meets with the substance. Maybe this is maltose. Let's pretend that this is maltose, and we've got fructose here and glucose. Then the enzyme can break the bond. Now these no longer, so our substrate for the enzyme was maltose. These are no longer maltose. They're now glucose and fructose, which means they don't fit the active site anymore. So they're let go and they float off. Notice the enzyme can still do stuff. It can do this again. It can move on and do this again and carry out the reaction again. The catalysts are not used up in the reaction. The products are now the products, they're different, but the enzyme is not used up. Okay, that's the basic essence from B1. Enzyme structure, it's a 3D structure specific to the substrate. The little thing about the names, so highlighter ace, basically the name often, not all the time. I would say not very much, but often, if you want to know what the substrate of an enzyme is, it's in the name. Annabolism and catabolism. Remember, cat is scratchy, so it breaks things apart. Catabolism is breaking things apart. So hydrolysis, remember we talked about hydrolysis in Unit A, that's breaking down polymers into monomers or smaller molecules. That's hydrolysis is an example of a catabolic reaction. Dehydration synthesis, you betcha dehydration synthesis is an enzyme-mediated reaction. That is anabolism. Catabolism releases energy. It's catabolic reactions. The net, so you have to have energy to activate the reaction, but then in catabolism, when these bonds break, energy is released, and the net energy is greater than the energy it took to start the reaction. That's catabolism. So it's exothermic. Usually these are exothermic. They increase, things get heated up. Anabolic reactions, when you put things together, the energy is absorbed to do this. So usually these are endothermic. They actually take up heat. Okay, and we'll talk about models in Unit B. So there you go. That's A1. Oh, it's only an 11-minute video. Yay!